I had the opportunity to meet him during my visit to the USA, and also I worked with him in a pharmacy college for few years. And I say thanks because because it's a weekday as well as with tight schedule in the industry, he could accept our invitation. Thank you, sir. And yeah, we are eager to listen to the session, sir, about clinical IND and NDA submissions. Thank you, sir.
Can I start?
Yes, sir, you can start.
Yeah, thank you. So first of all, I appreciate Dr. Mamata Madam for organizing this faculty development program. So and thanks for introducing me to the team. So it's a great opportunity. So whatever I'm going to present here, all the industrial experience. So for the last 3 decades, I was in the teaching for 10 years, and then industry. I worked in R&D formulation, and then I worked in analytical. Then I worked also in validation, manufacturing, and regulatory. So encompassing all the, so the cumulative experience, I would like to share my practical knowledge and everything. So it's a great opportunity, and as much as possible, I prepared around 45 slides. So basically, it's going to be help you in the the future whoever is going to join in the industry. I'm going to simplify everything. Please feel free to keep me posted for all any questions to my email ID or in the chat box or to Dr. Mamata Madam. In case if I don't get opportunity to take all of to take up all your questions, because in the interest of time, I would like to start my presentation. So, are you able to see my presentation?
Yes,
Are you
sir.
able
It's
to see?
clear.
Yeah.
It's clear.
So today I'm going to talk about clinical IND and NDA submissions. Uh, First of all, I would like to make a disclaimer. So the views in this presentation is completely the views of mine and my personal experience and in my personal capacity, and it does not reflect the views of the either current or previous employers. Whatever is presenting today here, everything is available in the public domain. But the only thing is I'm going to make it everything in a organized way. So it's a disclaimer. So first of all, in the interest of time, global IND and NDAs. So how the drug discovery start, pre-IND, NDA, new drug applications, and then to ANDA, of course. But my focus is on today on the regulatory aspects, right? So it's a global regulatory agencies list I would like to present here. So first of all, USA and Europe are the biggest markets, right? Everybody wants to focus on the INDs in these two regions, right? America, in USA, the regulatory agency's name is FDA, Food and Drug Administration, and Health Canada, HC, and Australia is a TGA. And of course, in Europe, there are centralized filing submissions called, and also mutual filings and country specific filings, EMEA, right? And MHRA UK. And of course, I will show you again in the presentation all the list here. One second. So the global regulatory agencies list, everybody wants to first of all target USA, and then after approval, everybody wants to go to the rest of the regions. US Food Drug Administration, one second, let me, FDA, European Medicines Agency, EMA, Health Canada, and PMDA Japan, Pharmaceuticals and Medical Devices Agency, Australian Therapeutic Good Administration called TGA, MHRA UK, Medicines and Healthcare Products Regulatory Agency, CDSCO India, NMPA called China National Medical Products Administration, South Korea Ministry of Food and Drug Safety, NVISA Brazil, HPRA Ireland. So like this, so each and every country has their own specific country specific regulations. Going forward, I'm going to see, explain you how the regulatory strategy needs to be built up, specifically when you're targeting to USA. China is a specific one where currently you know that there's a lot of constraints, you know, when you wanted to get approvals from in China, right? So I think country-specific guidelines with respect to USA, because it's a good market, is the biggest market. Most of the clinical trials, most of the NDA companies, IND activities will be going on in Europe and USA. So all these are global, you know, regulatory agencies list. And then this is overview. Today I'm going to complete, you know, cover in my presentation preclinical IND investigational drug, NDA new drug applications pathway, clinical protocol, clinical amendments. These are very, very important at phase 1, phase 2, phase 3, even for NDA, right? Chemistry, manufacturing control, CMC, and everything will be governed by the ICH, International Council for Harmonization, which is the really, you know, harmonized body for across all the geographies, for all the covering every, you know, nowadays even from the 2015 onwards, even China, East Asia, and every country is being covered. There is a harmonization for all across all the countries. So particularly ICH is very, very important, which we are going to talk about on the module 1, module 2, module 3, module 4, module 5, which is the real, you know, the compiling, drafting regulatory modules. And then I'm also going to touch base on the pharmacovigilance, safety data, DSER, drug safety, you know, development of safety update review, and the suspected, you know, adverse reactions, periodic adverse drug reactions, periodic benefit, you know, reactions. So all these are called safety update. This safety will be collected, the data will be collected and updated to the regulatory agencies periodically, right from the, you know, the pre-IND, and then IND phase 1, phase 2, phase 3, even NDA, even the life cycle post NDA activities. I'm also going to touch base on the change controls. That is how we do the regulatory impact assessment as well as quality impact assessment, which is the really important, you know, using the extensively the Viva Vault tools in the industry. And also I'm going to cover on the type of meetings, the type B meetings. These are the typical meetings what we have regularly with the FDA. Keeping so my focus is on today on the FDA pre-IND meetings, end of phase meeting EOP2, and pre-NDA meetings. How regularly, you know, we communicate with the agency FDA. So in order to address, you know, the clinical aspects, clinical protocol amendments, and the CMC manufacturing issues. So then only, you know, till the phase of NDA, it's a continuous dialogue and communication with the health authority in USA, you know, FDA. And I'm going to touch base, you know, just, you know, layout on the comparison of the regulatory on the different countries, how the comparison between the USA, Europe, and China. These are the major, you know, markets, right? Okay. Then with this overview, I'm going to straight away going to the explain on the IND. So this is called IND, drug discovery phase, you know, pre-IND. First of all, there is a, you know, drug discovery phase, right? You know, the chemical synthesis, chemical characterization of the active pharmaceutical ingredient, extensive, you know, data should be available. So then pre-clinical, pre-IND phase is very critical, where animal testing, extensive screening will be done, like genotoxicity studies, toxicology, general toxicological studies, carcinogenic studies, you know, like even for the pediatric, like, you know, the embryo fetal studies, the selection of animals, like, you know, rodents, non-rodents, like dogs, monkeys, you know, and then also mice, rats, rabbits, you know. So the selection of the animals will depend upon the disease, you know, the disease specific. So here this is very, very critical. Like any research bodies or research institutions or any university or any, like, you know, they can carry out the activity of the animal screening. So once you generate the data from the animal, you know, the studies, first to FIH, first in human studies will be conducted from the phase 1 IND. So preclinical, pre-IND phase is nothing but animal studies or toxicological studies. From the IND phase 1, phase 2, phase 3 will be the investigational new drug applications. The first the first in human drug will be administered in the from the phase 1 onwards. Everything should be GMP manufacturing activities, like phase 1 to phase 2 and phase 3. The main emphasis as we go from phase 1 to phase 3, mainly the the safety, and then as we go from phase 1 to phase 2, efficacy. More number of subjects, say for example in the phase 1, um, maybe typically 20 or 25 or 30 subjects, human volunteers, so human subjects will be enrolled. So maybe the subjects will be diseased patients or even healthy patients. That depends upon the protocol how you develop. But before going to the phase 1 here, pre-IND meetings here. So pre-IND meetings are very critical before going to proceed any phase 1 clinical studies. What are pre-IND studies? Because once you generate all the data, compile all the data in the form of module 1, 2, 3, 4, ECTD, electronic common technical document. I'm going to explain in my subsequent sections. Then whatever the proposed protocol design, the layout, general investigational plan, and who is going to conduct the studies, like investigators, the CV, and then IRB approval, that is called institutional review board. So in Europe, it's called as a institutional ethical committee. So all these approvals will be taken and present the data to the FDA during the pre-IND meeting. So in the pre-IND meeting, whatever you generated the data on the animal studies present, and whatever you are going to propose the protocol, everything should be laid out here. So once the FDA is convinced, then yes, fine, you can carry out and you can start the phase 1 studies. If the FDA is not convinced again, okay, hey guys, this is not acceptable, try to modify the inclusion criteria, exclusion criteria in the protocol, because that is very important for the study. Then again, they will revert back to you, to the sponsor, whoever is going to the sponsor, maybe a commercial IND or any research oriented, the sponsor or investigator, whoever. So they have to modify the protocol. This is called protocol amendment. So pre-IND meeting in the case of USA, CTA, clinical trial applications, in Europe, they call it as. So CTA Canada, CTA Europe, CTA China, all these are equivalent to the IND in USA. Okay. So once the phase 1 is done, again, you need to conduct the one more study called phase 1, maybe type A, type B meetings with FDA. Once you clear to generate all the data, like, safety pharmacology, pharmacokinetics, and toxicology, safety and efficacy, everything, you present to FDA. Then again, here also you need to get a permission before going to the phase 2 studies. So end of phase 2 studies is very critical. See, this is called end of phase 2 submission. During the end of phase 2 submission, whatever you have conducted and generated all the data from the toxicological studies, phase 1 and phase 2, you present to the FDA. And again, whatever you are going to propose to the phase 3 studies, the layout, everything you have to present to the FDA. Okay. Once the FDA says yes, then you are allowed to carry out the phase 3 studies. Phase 3 studies, the main emphasis on the efficacy. Efficacy in a larger population, maybe 1,000 or 10,000, whatever, So as we go from phase 1 to phase 3, increase the number of subjects. And at the same time, we also monitor the tolerability and also safety. In all these phases, collect the safety data as well, efficacy too, and present to the FDA before pre-NDA, called pre-NDA meeting is very, very critical. So here I mentioned MAA means marketing applications, and BLA is biologics. Biologics are mainly, the peptides, proteins, or vaccines. And typically today I'm going to focus on the small molecules. Okay. So before, for the NDA, there is a huge requirement. That is, manufacturing of 3 registration batches equivalent to commercial size or even less in the case of rare disease molecules. And by the time we come to the NDA, everything should be established, like, CMC, your drug substance, drug product, complete development, complete validation, specifications, everything should be locked and well defined and established, all the things. Then you present to the FDA, hey, this is the 3 registration batches data. These are the stability studies, particularly accelerated stability studies, 6 months data. Long-term studies may be around 12 months for the typical NDA, right? Present everything. The content will be the module 1, module 2, module 3, module 4. Then if the FDA says if it is accepted, then you're going to market the NDA to the, to the commercial market. So all these things will be the innovative drugs. The branded drugs, typically they carry out all the activities actually. Okay. So once the approval from the NDA, the branded companies, right, if anybody wants to join, enter as a generic company, so there are called as a paragraph 1, paragraph 2, paragraph 3, paragraph 4. There is a different subject. So generic companies are not innovative companies. Typically, the branded companies, they will do extensive safety and toxicological and efficacy evaluations. They get approved to the NDA. So if it is a new molecular entity, they call 505, 501B1, 501B1 pathway. So this is the overall layout. So from NDA to IND to NDA, biological applications to again NDA, abbreviated new drug applications, and then OTC. So this is the overall drug discovery, to the NDA pathway. So if you have any questions, you can comment. So with this, I'm going to, the next slide will be the preclinical toxicology IND. Preclinical toxicology is a critical component of the drug developmental process. The goal of toxicology testing is to support the transition of a new drug candidate to patients in the phase 1 clinical studies, as I mentioned earlier, through investigational new drug filing. The IND application consists of the information on the preclinical safety and toxicology, chemistry and manufacturing to support the initiation of the clinical trial. So it contains the complete preclinical toxicology. IND content will be animal pharmacology, toxicological studies to assess that to assess the new drug product is safe for initial testing in humans. Manufacturing also is very important. So during the initial phases of the phase 1, phase 2, clinical is really the important factor for the agency, for any sponsor. As we go CMC, how we are going to manufacture the drug characterization, drug substance, and the drug product, which I'm going to show in the subsequent chapters. Okay, slides. The toxicology studies includes the how the toxicology studies in the human in the animals will be single dose, repeated dose, reproductive, developmental studies, and the genotoxicity studies. These are very, very important in the beginning, because even the drug substance, for example, the degradants, the metabolites, whatever, coming from the drug substance, they may cause, genotoxicity or carcinogenicity in the animals. So we have to evaluate upfront before going to the IND phase 1. So everything is laid out how exactly you need to carry out these studies, including bioanalytical method validation and everything as per ICH guidelines. Okay. Please let me know if I'm going very fast and slow and everything, So here again, I'm going to lay out on an investigational new drug application is a request for authorization from FDA to administer an investigational drug or biological product to the humans. An IND must be authorized prior to the interstate shipment and administration of any new drug or biological product that is not subject to an approved new drug application. So, okay. So the INDs will be the 2 types or 3 types, because one is commercial, because of if the companies wants to go for the IND meant for the market, is called as commercial. Any research oriented institutions like non-commercial, they submitted mainly to advance the scientific knowledge. The INDs can still be submitted by a paper or even or by an e-CTD format. So this is the overall scenario, how the drug discovery from the pre-IND, the animal studies to the phase 1, phase 2, phase 3, the overall pathway. So now I'm going to talk about ICH. ICH is a paramount importance, and everybody wants to, follow the guidelines which is mentioned in the guidance documents. So basically, QSEM. Q means all quality guidelines. S means all safety guidelines, which I'm going to talk in the next slides. And efficacy means E means efficacy and multidisciplinary guidelines. So as I mentioned, preclinical, mainly toxicology, the emphasis, you have to follow all S guidelines. And module, all the clinical phase 1, phase 2, phase 3, all the clinical data will be, in the module 5, that is called efficacy. To what extent is your drug molecule is highly efficacious? The rate and extent of drug absorption, bioavailability, pharmacokinetics, toxico, the receptor drug, binding reaction, and ultimately how it's going to elicit the pharmacological activity. So there are primary endpoints, pharmacological primary endpoints, secondary endpoints. There are so many models to assess the efficacy and everything, right? So typically, so toxicology will be here, and the safety in the humans, all the phase 1, phase 2, phase 3 will be here, and of course the drug substance, and the drug product will be covered in the quality guidelines. Okay, this is the overall gamut of the the layout of ICH. So what is the regulatory landscape? As I mentioned, how the industry will be divided, like first of all, in the departments like a non-clinical phase, clinical humans, CMC means manufacture, CMC chemistry manufacturing controls, which contains all the manufacturing and characterization specifications, analytical measures of the drug substance as well as the drug product. Of course, quality is very, very important. Ultimately, you're going to file to the health authority. Health authority is nothing but all the regulatory agencies across the world, whatever I have mentioned in my first slide. Okay. Then now I'm going to talk about expedite the drug development for serious conditions of filling unmet medical needs. There are a kind of, like a fast track pathway, breakthrough therapy, accelerated approval, priority review. For example, in the for the last 4 years ago, maybe because of COVID pandemic, because of the pandemic, COVID vaccine was approved under the emergency use of authorization, because that's very important, because in order to meet the unmet needs, it's very under emergency basis. Likewise, there are so many rare diseases, orphan drugs, which are unmet medical needs. Fast track will be is a process designed to facilitate the development and expedite the review of drugs to treat serious conditions to fulfill an unmet medical needs. Likewise, if there are existing therapies are not available, substantial improvements you wanted to make, you can file under breakthrough therapy, called as a under special designations. So because the FDA will be giving a lot of subsidies or whatever we call as waivers. You can bargain. Hey guys, I'm going to working on the fast track therapy because it's a very unmet medical needs. I wanted to file as soon as possible. Can I reduce the number of patients, or you want to have a long term safety data? So a kind of negotiation you can work with the FDA with all the type A, type B, type C meetings. So these are the main special designations where one can submit to the agency. This is very important. This is a real core part of IND. How you are going to file any submission, any documents, compilation of all the documents, drafting, the submission process, contents, this is a very typical part. So for whatever I have mentioned, that is a strategy, technical know-how. One has to keep that one as a background to write the drafting and everything, all the module 1, module 2, module 3. As I mentioned, module 1 is only a administrative documents, like a cover letter, which I'm going to show it here. Form 356, Form 1571. What is Form 1571? I'm going to show it here actually. Form 3674, clinical trial certification, clinical supply labels, general investigational plan layout, how you are going to do, how many number of subjects, whether it's a randomized subject or crossover technique, so phase 1. So what complete layout and even protocol and investigators brochure is very important. It contains a whole list of complete pharmacology, drug metabolism, complete historical data of everything about the drug. We have to capture in the investigators brochure. Whenever there are any updates, we need to update in the investigators brochure, right? Module 2 and module 3 will be the chemistry manufacturing controls, like how you are manufacturing the active pharmaceutical substance. For example, drug master file. That is called, the API as such, called as the CMC and the drug substance. When the drug substance combined with the excipients called as a tablet, it is a dosage form that is called as a CMC and the drug product, which I'm going to show granularity. This is the high level layout. I'm going to show in the next slides on the granularity. Likewise, module 4 will be all the data, all the reports will be captured, and it has to be reported in the form of a module for non-clinical reports, pharmacology, toxicological information, non-clinical reports, particularly animal studies. And module 5 will be the human clinical studies. So all the protocol, first of all, how you are going to submit to the IND. And Form 1572, investigators curriculum vitae, their qualifications, who is going to carry out the clinical trials and everything, and clinical reports once the data is, once the study completes, then the data log, statistical uh, analytical plan, and then you have to make a CSR clinical study reports. Everything will be captured in the form of module 5. So module 5 is a human clinical studies. Module 4 will be non-clinical toxicological animal studies. Module 3 will be the CMC. The summary of module 3 and 4 and 5 will be captured in the module 2. Quality overall summary of CMC, summary of toxicology, summary of pharmacology and pharmacokinetics, and a summary of clinical studies. So this is the high level layout. I am going to show at granularity level. This is called, the real, how you are going to start the drafting. For example, they mentioned module 3. As I mentioned, module 3 is a drug substance, right? Or drug product. So in the case of module 3, S means drug substance, P means product. Okay, 3 to S1, general information, nomenclature of the drug substance, your API, your structure, your general properties like pKa, solubility, all these things will be captured here, like 3 to S2. So you are going to capture everything, the data, and all the reports drafting will be done as per the ECTD format module 3 for drug substance in the 3 to S. Likewise, manufacturers, who is going to be, what are the sites of the manufacturing locations, their address, their DUN numbers, DUN numbers, their establishment numbers, then description of the manufacturing process, what is the synthetic route, in process controls, intermediates 1 and 2, regulatory starting materials, all these things will be captured in the 3 to S22. Likewise, 3 to S23, control of materials. What are the specifications, for example, certificate of analysis. Likewise, 3 to S24, control of critical steps, and 3 to S25, process validation. Once you develop, complete the drug substance manufacturing, you go to the next level of validation. The validation is one of the important criteria that is equivalent to the commercial level scale. When you go to the scale up level, you're going to make some changes. Everything will be captured. If any changes, that has to be captured by using a change controls or a change management. Likewise, 3 to S3, characterization in like structural illustration. Like once you develop like, NMR, mass, LCMS, so impurities are very, very important. Whatever is coming from the drug substance is being carried over to the drug product also. You need to control, you need to have a, specifications at the release specifications or stability specifications of the impurities for the drug substance. Everything will be captured here. What are the main impurities, whether these are the degradants or process impurities, what are the limits you are going to establish based upon the ICH guidance, that is called identification threshold, qualified, threshold limits. There is one guidance is available in the ICH guidance as per the maximum daily dose. Likewise, 3 to S4, control of drug substance, I'm going to show in the all the, in my next slides. 3 to S1 specifications, 3 to S4 to analytical procedures, like, SA, the, complete characterization, their name and who is a manufacturer, vendors, and 3 to S4, 3 validation, analytical procedures. So everything will be the here, all the complete granularity. You have to capture everything. Remember, so when you're going to submit to the FDA, for example, one NDA is going to be list of 10,000 or 20,000 papers, pages. For example, it's contain extensive, comprehensive list of all the, the data, whatever you generated for right from the pre-NDA to the NDA. So likewise, I'm going to show it here. 3 to P means product, is a tablet. So 3 to P1, description and composition. Only you need to mention this particular section here. Likewise, 3 to P2, pharmaceutical developmental name, components, what are the components, like, like acetaminophen or hyperamellose or microcrystalline cellulose or dibasic. So you need to mention what the formula composition here, what are the IAG limits, inactive ingredients guide limits, everything will be captured here, Likewise, drug product name, manufacturing process development. What is the manufacturing process? It's a wet granulation or direct blending process in the case of small molecules, or roller compaction. Why you have selected? How did you develop, by using a QBD, quality by design, all in process, process controls, evaluation and optimization of the process controls, everything will be captured in the 32P23 section. Likewise, 32P24 is container closure system. Microbiological attributes, compatibility, drug excipient compatibility report will be captured in the 32P26. Likewise, 32P3 manufacturer. Who is the manufacturer? Where is the manufacturing site of the drug product? The location, address. Who is then what about the packaging, the activities, the location. So batch formula, what is the batch formula? Everything will be captured. These are granularity, this is a typical e-CDD format of module 3. One has to understand and compile all the data, and the drafting will be in the e-CDD format. Please let me know. I'm not seeing any. I think the charts, I'm going very slow or fast. So please let me know. Accordingly, I can modify my this one. So as I mentioned earlier, again, IND submissions, phase 1, phase 2. These are the typical, as I mentioned, 1571. Let me show what is 1571. So this is 356 form. 356 form is just a fillable form to the FDA. It contains, applicant information, application to market of a new or abbreviated new drug application or biological for human use. What is who is the sponsor or manufacturer, or who wants to, to become the marketing holder, telephone number, applicant address, authorized USA agent, product description, NDA or ANDA or biological application, establishment name, proprietary name, chemical name, dosage form, all these things will be captured here. Application here, whether it is a new drug application or abbreviated new drug application or biologics application, whether the NDA is 505B1 or 505B2. 505B1 is a new chemical entity. It's a completely pure new molecular entity. Nothing is existing in the market. You are developing and you are finding out and you are, filing as a NDA. 505B2 means that, for example, you have a drug product of, say, for example, and 5 milligram and 10 milligrams, you wanted to develop another 20 milligrams, is a line extension. Existing, if any modifications, you have to file under 505B2. Likewise, what is the submission criteria? Whether it is original or you are going to make a efficacy supplement or any modifications, that submission, or it is a post-marketing. Once after the NDA approval and the life cycle management, you are going to make any changes post-marketing requirements, REMS, risk evaluation mitigation statements and all. So this is the typical 356 form, fillable form. It's very easy. So whoever is going to working in the regulatory, they have to, fill all the 356 form. Likewise, this is a 1572 statement of investigator. This is very important. Investigator, whoever is, under whose uh, uh, jurisdiction all the clinical activities is being performed. He is the responsible person. So we have to submit all the statement of the investigator, their educational qualifications, curriculum vitae, their address, where he is going to carry out the clinical studies, their location, and financial disclosure, that is not having any financial interest, kind of stuff in the, in this study. This is called 1572 form. Okay. Likewise, I'm going to show 1571 investigational new drug application IND 1571, as I mentioned. So all these forms are, one has to fill and in the module 1 as per eCTD format. Who is the sponsor? Their address. Okay. then list everything, initial investigational IND application, request for a reactivation or reinstatement, or if it is a developmental safety update. So is it typically is a fillable form, is a phase one study or phase two. You wanted to submit any filing in the phase three, you tick mark here, and this is the format. So 1571, 1572, 356, of course. There is a general cover letter. So these are the main module one documents. So as I mentioned in my first so in my previous talk, I mentioned that clinical protocol is one of the important document one has to design uh, from the phase one, in the phase one, phase two, phase three till NDA, right? This is very important. So it depends upon your therapeutic areas, oncology, depression, anti-depression drugs, respiratory drugs, like oncology, like lung cancer. So whatever the as per the disease therapeutics, one has to come up with a criteria design. The protocol is the really hallmark of any study. For example, this is the document available in the public domain FDA site. I just pulled for our illustration, Boehringer-Ingelheim document. So BI Boehringer-Ingelheim trial number and investigational product number and the title. So pharmacokinetics, safety, immunogenicity and efficacy, Humira. What is Humira? It's all rheumatoid arthritis actually in the pain conditions, inflammation in the treatment. How you are going to design? So clinical phase three. Likewise, what is the design? What is the title of the trial? Clinical trial protocol, pharmacokinetics, safety, immunogenicity, and efficacy of the this compound versus Humira. You're going to compare this one study with the branded one already existing like Humira in patients with the moderate to severe chronic plaque psoriasis. So the criteria, the design is that you're going to carry out a randomized trial in order to prevent the bias. Double blind. Double blind studies means that either neither the investigator nor the patient, they don't know what drug will be given to the patients actually. We have to blinding study to prevent the bias. Likewise, parallel arm. What is parallel arm? So the main difference is that because those drugs which are having a the longer half life, so they cannot be they cannot be done by using a crossover techniques. They can be studied only under parallel studies in order to, because they have a longer half life. So by knowing the clinical, the condition, and one has to come up with a clear cut design and get it approved by the agency. Multiple dose active comparator trial. So you're going to make a one comparator that is called control one. So likewise, trial site, multinational. You are going to study not only in USA, maybe in Europe, multiple site, multi-center, because one has to address multi-ethnic groups, diversity of population, diversity of age groups, and everything. So that's why they mentioned the study will be conducted in a multinational, multi-center trial, approximately 80 clinical sites across the approximately 12 countries. This is phase three. Phase three, because phase three, we are going to establish long-term efficacy data, extensive pharmacokinetics data, and that is why the study will be conducted in multinational, multi-center studies. So I'm just showing this is the typical protocol. So number of patients entered, enrolled, and what is the main inclusion criteria and exclusion criteria. That is very important here. Actually, how you are going to carry out. So for example, inclusion criteria means that if why what is inclusion criteria? In this protocol, whoever wants to, the disease conditions patients, for example, the males and females agreed greater than 18 years of age, less than 18 years of age at the screening who have diagnosis of the moderate to severe chronic plaque psoriasis. So this is the inclusion criteria. So you have to enroll the patients only within this age only. Likewise, exclusion criteria. For example, patients with a significant disease other than the psoriasis or any other significant uncontrolled disease they cannot be enrolled in this study. They come under the exclusion criteria. So just I wanted to say how the design will be conducted for the clinical trial protocol. Likewise, as I mentioned, investigators brochure, physical, general investigational plan, how the NDA document will look like. This is the, Center for Drug Evaluation Research, clinical reviews, 505B1 application NDA, NDA number, priority review. Okay. Then what is the division in the FDA? Complete list of all the data will be captured in the NDA. So I think this has been already covered. So ICH M3 guidance will be pharmacology, reproductive toxicology to assess the reproductive safety of the drug after repeated and chronic exposure, segment one fertility and early developmental to implantation, definitive embryo fetal development. Segment two will be fetal, neonatal and peri-postnatal, genotoxic studies and general toxicology. So with this clinical, I think I have shown already the clinical trial protocol, how the design will be conducted in the phase one, phase two, phase three. Typically, the template will be more or less will be the same. Okay. And then now, what are the ICH guidelines? International Council for Harmonization across the geographies, all the countries will follow the guidelines. Quality, as I mentioned in the earlier slides, safety guidelines. E means efficacy. M means multidisciplinary guidelines. The safety guidelines are, S1 to S1C carcinogenic studies, genotoxicity studies, toxicokinetic and pharmacokinetics, toxicity testing, reproductive toxicology, and then all the studies will be captured under safety guidelines. This is M7 module. M7 is the assessment and control of the DNA reactive mutagenic impurities in the pharmaceuticals to limit the potential carcinogenic risk. Say, for example, recently because of nitrosamine impurities, there's a lot of issues across the world. That's why the CEDAR, FDA has issued a new guidance called nitrosamine impurities. Metformin is one of the anti-diabetic drug. So from now onwards, every new molecule, they need to assess the nitrosamine impurities. Is a primary, secondary amine, which is causing the nitrosamine impurities, they cause carcinogenic, they are carcinogenic and even genotoxic. I mean, nitrosamine impurities are mainly carcinogenic. For this, in the FDA is keeping a more emphasis on the even toxicological animal studies, that spiking the metabolite or in degradant or nitrosamine impurities after isolation, inject or spike into the animals to see the how far extent they are causing, or to evaluate the carcinogenic activity. At the same time, bioanalytical methods validation is also one of the requirement. Even right from the phase of preclinical, FDA wants to see to what extent the analytical method development and validation. So safety guidelines, as I mentioned, carcinogenicity, genotoxicity, toxicokinetics and pharmacokinetics, toxicity, reproductive toxicology. So these are all safety guidelines. And uh, genotoxicity and carcinogenity in the animals studies. Then efficacy. Efficacy means that in the humans, clinical safety for drugs used in the long-term treatment, pharmacovigilance safety, clinical study reports, complete compilation of all the individual as well as summary reports, dose response studies, ethnic factors, good clinical practice. So from E1 to E12, everything will be efficacy studies. So I think with this, I captured everything like what is ICH guidelines, safety guidelines, efficacy guidelines, quality guidelines, multidisciplinary guidelines. Then I'm going to talk about the CMC. CMC is very important because of the drug substance manufacturing, drug product manufacturing, all the the data will be captured as per the quality guidelines mentioned in the ICH. Q1 to QF stability. What are the stability requirement? There are many zones: zone 1, zone 2, zone 3, zone 4. Typically in FDA, they want three batches data, 6 months ACC conditions, 40, 75, 40 degrees centigrade and 75% RH humidity conditions. Intermediate maybe or long-term stability is also very important. Analytical validation. During the course of the, as you move from phase 1, phase 2, phase 3, parallelly, clinical study is in progress. At the same time, CMC also development will be in progression. So the development after developing the analytical methods, you need to validate the analytical methods. For example, linearity, specificity, all those things in the criteria mentioned as per the ICH guidelines. Impurities also is very important. Like, ICH, impurity, how you are going to come up to the specifications in the drug product or drug substance, we have to follow the guidance Q3A and Q3E. Q4 pharmacopoeias, Q5 quality biotechnological products, Q6 and Q6B is specifications, For example, content uniformity, SA 95 to 105, why, how? So likewise, good manufacturing practice, GMP, pharmaceutical development, everything will be captured here. How you need to develop as per the QBD, right? Quality by design, by using a design of experimental trial batches, everything. So everything will be mentioned here, captured as per in the Q8. Likewise, Q9 quality risk management, pharmaceutical quality system. So this is the overall ICH quality guidelines. So now, so far after the clinical studies, as per the today IND, clinical IND and NDA, just I wanted to touch base, though it is not part of the topic, but I wanted to touch base on the CMC manufacturing. Typically, quality guidelines are, address all the chemistry manufacturing. Like, for example, this is the typical formula composition. This is an active pharmaceutical ingredient. Okay, all these are excipients. Say, for example, these are rate controlling polymer. If you mention this is the formula composition, this is the percentage of W by W, of the composition. You need to know what will happen when you vary the conditions of the rate controlling polymers. If it is 13, what will happen if it is 8%? What will happen if it is 18%? So by with varying conditions of these excipients, which are very critical, how the impact on the critical quality attributes. What are the critical quality attributes? Like, content uniformity, dissolution is the one of the important critical quality attribute. So you need to come up with what are the CQA, critical quality attributes. Likewise, all these are formulation variables. Individual material attributes also you need to come up as a risk variable. This is a typical quality by design, diagram. So if it is a direct blending during the process, say for example, when you are mixing all the excipients with the API using a direct blending process, you are blending for 10 minutes. What will happen when you mix 8 minutes or 12 minutes? A kind of design space. So process development and process optimization, formula composition development and optimization. So these are the prerequisites for the QBD, quality by design concepts. In the early phases of the development, you need to apply the QBD. Once you come to the commercial level validation, it has to be in the risk-based approach. You need to validate all the process parameters, maybe in process parameters or final, the end process parameters. So this is the typical, both formula composition and formulation optimization and process optimization. So you need to, this is the typical QBD. This is an API particle size. If you change the drug substance particle size on the drug product, you mentioned the risk is low. Why it is low? You need to come up with a justification. For example, in the case of direct blending process, when the particles are 100 microns or even 500 microns, okay, how the impact on the dissolution are on the process. You need to make a upfront risk evaluation, low, high, medium. So once you say that after making this design of experimental trial batches, you need to evaluate. Finally, you have to come up with why it is low or high. So this is called QBD risk-based approach of all material attributes and how ultimately is going to impact on the critical quality attributes of the drug product. So this is a typical chemistry manufacturing controls. Main thing is QBD, and the second one is the risk-based approach. And module 3 will be capturing all the chemistry manufacturing controls, both for the drug substance and the drug product. So with this, the regulatory requirement, how the phase 1 NDA, phase 1 IND, phase 2, phase 3, how the clinical study program, how the CMC program will be carrying on, we have already discussed. Now I'm going to compare the comparison of the between the IND, NDA, BLE. So typically, IND will be filed, preclinical. They need, as I mentioned, preclinical data, CMC, proposed CT, I think protocol types, maybe investigator initiated or emergency treatment. NDA will be under two rules 505B1 and 505B2, as I already mentioned. So ANDA means generic drugs. The generic drugs are nothing but the copycat. I cannot say copycat because NDA holders will be conducting extensive safety and toxicological and efficacy data, but only the ANDA people, the generic drugs, they just to show the equivalency between the equivalence with the comparator like all, RLD or NDA drug. So they follow typically 505J, and if it is biologics, they call biosimilars. All these are same I have already mentioned. So this is a high level regulatory strategy which may not be required for the talks, but people who are working in the industry, generally, the strategy is that basically US is the biggest market, but China till 2015, there's a lot of gaps. For example, lot of delays in getting approvals in China in NMPA because focusing on the clinical trial strategies, bridging trials and multi-regional clinical trials are typically required by the NMPA in East Asia, resulting in longer clinical delay time, whereas EMA, that is Europe and Japan, primarily require international MRCT, that is multi-regional clinical trials in Europe and in America, with a clinical delay of less than 5 months. So that means that there's a gap, slight significant gap between the clinical trial durations between China and other countries. Say, for example, when you get approval in USA, you cannot use the same dosage in China because China needs a kind of clinical study which should be carried on in the Chinese mainland because they wanted to address the ethnic sensitivity study because some of the populations which are in the, say, for example, in the case of lung cancer, when you conduct all the studies in the multi centers in USA and submit to the FDA, right? After getting approval, when you want to submit in China, they may not accept because the studies whatever you did in US, they may not address all the population in China. So you need a kind of bridging studies. That is why in the beginning stage, in the early phase of the development of phase 1 and phase 2, phase 3, you need to know the country specific guidelines like China, and also you need to build in the strategy in the upfront. So that way we can address even Chinese specific regulatory requirements where a kind of ethnic sensitivity analysis, because they wanted to address the kind of population, the same mitigation and safety and efficacy data should be available to the Chinese population also. If not, they will ask, hey guys, you are submitting a USA dossier to China. It may not be acceptable. You need to also show a kind of bridging study, a kind of a small population in Chinese, in the country of China, conduct a kind of clinical study, a kind of, bridging studies in China, then only we can accept it. So it is a strategy actually. So IND assessment teams. So what are the investigation new drug assessment teams are clinical in the FDA, clinical pharmacology, like, microbiology, statistics, pharmacology, toxicology, CMC, quality, chemistry, manufacturing controls, then policy. This is the IND. NDA will be the drug substance, drug product, biopharmaceutics, drug product process, facilities, microbiology. After approval of NDA called surveillance, inspection, of course, by audits. These are the typical departments in the NDA, like in the FDA, right? Then, I'm sorry. Then meetings. The meetings, communication and FDA meetings are very important from the phase of preclinical, phase 1, phase 2, phase 3. These are called as a type A meeting, type B, type C, and type D. They just, to negotiate with the FDA, whatever you generated the data so far in the, say for example, the phase 1, when you wanted to go to phase 2, propose a protocol, clinical trial design, and get it accepted by the FDA. So a kind of, the continuous constant interaction and dialogue with the FDA during the course of all the phases of phase 1, phase 2, phase 3, and even after NDA. Typically, a type D meeting will be if you have one or two questions. Say, for example, you have some lot of questions like whether I need to do the validation of drug substance manufacturing or drug product manufacturing like 2 batches because I don't have any data. Typically, they require only 3 batches. You have a lot of questions. So try to give a briefing package, okay, communicate to the agency in the form of type D, one or two questions only. Type B will be the extensive, complete briefing package is very important where you need to capture what all the questions, 10 questions or 20 questions connected to CMC, clinical, non-clinical. Okay, I don't have this, I have this. Can I go with to the next level? So you will try to negotiate with FDA by using type A, type B, type C, and type D meetings. Okay. In the process of when you're moving from the phase 1 to phase 3 and NDA, there are two important things called change controls or change management. What are the change controls or change managements? When the FDA is asking you to modify say, for example, any clinical protocol, you need to amend the protocol. Or if not, if you are making any drastic changes in the drug substance or the drug product, FDA will ask you, okay, you are making a substantial changes or significant changes, you need to file an amendment and wait for 30 days. So if you don't hear back after 30 days, you can go ahead and proceed to the next level study. So this is a kind of, how the changes will be captured and how to address by using amendments. If it is a post-approval life cycle after approval of NDA, there are 3 categories. Whether the change is a major change or minor change or, moderate change. During the course of the phase 1, phase 2, phase 3, there are only two. One is major and the second one is minor. The minor changes are maybe in the protocol or in the maybe it is a CMC manufacturing or whatever. You don't have to do any permission, get permission upfront from the agency. You will be capturing in the life cycle annual reportable. If it is a major change, you should not conduct any study. You wait for it and you communicate to the FDA, wait for 30 days. If the FDA says that, hold it, they call it clinical hold, they call it as. Okay. These are the main, the change management or change controls. One has to come up with a regulatory impact assessment and change control assessment, which I'm going to talk in the next slides. So these are the typical, I think I have already discussed this one. Say, for example, type B meeting. Once you make a request to send the communication to FDA, the FDA will respond within 14 days. Okay. Then FDA received a meeting package. No later than within 15 days, you need to prepare a briefing package. What is briefing package? So far, whatever you have generated the data on the clinical studies, non-clinical studies, and formulation development, drug substance, and the drug product, you need to capture everything as per, in a structured format and submit the briefing package before that. And FDA preliminary responses will be no later than 5 days before the meeting. Requester response to FDA preliminary response will be no later than 3 days. So FDA schedule a meeting date will be within 70 days. Once you make a request here, within 7 days, they are going to give the, you know, give their consent, what is the minutes of meeting, what is the outcome. So this is after 30 days. So typically type A, type B, type C meeting is all communications with the FDA. It depends upon the how many number of questions you have it, like type D, only 1 or 2 questions. Type B will be typically end of phase 2, end of pre-NDA meeting. You, so this one you have to follow. So the all type B meetings. Now, change management with this, I'm going to, I think, wind up because already it's for time. Change controls are very important. What is change control? Whatever you have submitted to the agency, okay, in the module 1, 2, 3, 4, whether you are following the same process or specifications or the same methods, if you are making any changes on the, compared to the whatever you have filed or submitted to the health authority, then you have to initiate a change control. Particularly, the quality assurance will be initiating the change controls. But the regulatory people will do the regulatory impact assessment. As I mentioned, for example, I'm going to give one demo here. So generally, Viva Vault, it is a tool wherein one will do the regulatory impact assessment. I'm going to show it here in one demo. Mm. So this is the typical Viva Vault actually. So Viva, so how the home Viva Vault submissions like, Viva RIM. So it is a tool, regulatory information management. So the compilation, as I mentioned, in the ECTD format module 1, 2, 3, 4, one can initiate the drafting by using a Viva Vault. And also the communications between the FDA and the sponsor also can be captured, in the health authority interactions, the library, registrations, applications, products names, and everything will be, all these will be captured in the Viva Vault. It is a tool industry will be using extensively. So generally, regulator will be using this way. There are clinical people who will be using their own, customize the tool accordingly. Medical affairs also in the NDA, particularly medical data, medical license. So all these things will be, they are capturing, they will customize the Viva Vault, as per their needs. So here they mention investigational application number, application details, publication details, regulatory objectives, submissions, therapeutic indications, countries, everything will be, by using Viva, what is going on, what was there already, which sequence is there already. Whenever you wanted to update it with a new sequence, you can, what is going on. So you can know what to, when to update the sequence, in the site. See, this person has given an investigational submission 007 was the sequence. Now he's going to amend the protocol because the FDA is asking to modify the some of the content in the protocol, clinical protocol. So the sequence will be the next one. This is 7. The next sequence will be the 8. So submission name, protocol amendment, application number, application type, submission type, original application, submission subtype, amendment, new investigator, because of sometimes when the clinical phase is going on, the investigator may be, he may not be willing to do in the, say for example, some people, some investigators, they might have died. We don't know. So they wanted to replace a new investigator. So that time they wanted to file, the sponsor will file a amendment, protocol amendment. So this submission will be connected to that one. What is the dossier format? ECTD, Electronic Common Technical Document. Planned submission date, actual submission date. So this is a typical Viva Vault RIM, regulatory information management. As I mentioned, cover letters, US regional clinical study reports, literature, administrative information module 1, So now I'm going to talk about the post-approval changes. Once you submitted the NDA, you got approval to NDA, commercial market, everything is going on. But as I mentioned, that change management, whenever you want to make any changes, like, there are three categories. For example, one company has already, they got approval on one product A. If they wanted to add one more site, manufacturing site, because of the marketing needs, okay, that comes under a major change. Whenever you wanted to delete a blend uniformity from the existing approved filing, that is a major change. But whenever you wanted to make a, for example, you filed with the 30s bottle count stability to the NDA, which is already approved one. But when you wanted to add a 60s count bottle to the commercial market, whenever you wanted to execute or, to the launch, that is a moderate change. How you are going to, do the impact assessment, whether it is a major, moderate, or minor. So I will discuss in my next one case study. So all the major studies will be the prior approval supplement. In USA, it is called as a tell and do. First of all, inform to the agency, get approved, wait for maybe 4 months or 5 months. Once after getting approval only, you need to implement the changes. Corresponding to the major change in USA, in Europe, it is called as a type 2 variation. In Europe, I mean, kind of stuff, but they are given a different name called type 2 variation. So if it is a moderate change in the USA, post-approval changes, not in the phase 1, phase 2, phase 3, after NDA approval. So this is called changes being affected, CBE 30, moderate change. Okay. So corresponding to this, the European filing called type 1B. First to tell, wait for 30 days. If you don't hear back, you can go for the launch. You can implement the changes. It is a moderate impact. Likewise, minor, annual report, because whenever, every year you need to update all the changes, whatever, as per the change controls and the changes you made to the agents, maybe in the drug substance or the drug product, clinical safety update, and everything you need to update annually, called annual report. Okay, this is called type 1A. So do and tell. First, to do means implement. No need to get any permission from the health authority. Implement the change, and but at the end of the annual cycle, you can inform to the agency in the form of an annual report. Module, I think module 1.13. So this is what. So all these are governed by the change controls. The impact assessment is one of the important. So the only two aspects one has to know whenever if anybody wants to come to regulatory, all the content module 1, 2, 3, 4, how you need to make a drafting, as per the granularity, how you do the impact assessment. These two are the prerequisites for the anybody who wants to come to the regulatory. Before going to the pharmacovigilance, first of all, I would like to go to the case study. So I'm going to show here one case study. A manufacturing company obtained approval NDA under 505B1 because this is a new molecule entity, NCE. The sponsor wants to delete the blend uniformity test from the in-process control specifications from the commercial batches. How do you proceed, and what is your regulatory strategy and impact assessment? I can say it's a major change. If somebody says that it is a moderate, if somebody says it's an annual report, but it is a major change because blend uniformity, say for example, you need to know what is the product, whether it is a IR or modified release, you need to know. Blend uniformity means that whether it is a granulated product or direct blending process. Say, for example, by knowing all these things, if it is a direct blending process, before going to the tableting or compression to make the tablet, blend uniformity is one of the critical in-process control, critical in-process specifications, which you cannot remove it because you have already submitted to the agency in the form of a registration batches. You proved it, but when you go to the commercial batches, you need to keep the blend uniformity also in all the batch records. But if you wanted to remove the blend uniformity, do the impact assessment, It is a critical in-process control specifications in the for the product. It is a major change. Okay, if you wanted to delete the blend uniformity because of commercial batches, is going to economize safety economy purpose, to save the time. Say for example, establish the data for 1 year or 2 years. Establish the commercial batches data for a blend uniformity, maybe 10 or 20 batches. Make a statistical analysis. Hey, I just, this is the data. I wanted to show that all the data looks very good. Can you please recommend, just I wanted to recommend, suggest to remove the data for the upcoming batches. Because this is a major change, file for the major change under prior approval supplement. So what type of documents you need to submit to the agency? First, in order to, to blend uniformity deletion from the existing specifications, which is already approved one, you have to file prior approval supplement. So how you are going to submit cover letter? Okay, blend uniformity comes one of the in-process control. As I mentioned in the beginning that it is one of the 3 to P3. I think there is one more called as a critical process of parameters. That particular document you need to replace it. Okay, update that one, and also batch records. Show all the data. So whatever the respective impacted sections, not a whole list is not required, only the respective impacted sections in the module 3 only you need to replace and submit to the agency, and wait for 4 months. Let them review and come back. If any questions in the form of information request, if they wanted to ask any kind of data, you submit it and get approval. Then only after approval you can implement the changes, and you can delete it by updating the batch records. So this is a major change. So this way how to do it, because blend uniformity is one of the critical parameter which will be impacting upon the critical quality attributes in process parameter. Likewise, a case study where you wanted to just only update the batch records. Batch records only just administrative things only. No need to update, no need to get any permission from the agency. You can update that one in the annual cycle, annual report. That is module 1, 1.13. So you need to know which section in the module 1 will be impacted. That one you have to update to the agency. So do the impact assessment as per the guidelines, update the respective impacted sections to the agency. This way, how to do the regulatory agency strategy. So I wanted to show first now pharmacovigilance. Pharmacovigilance is one of the important aspects which will be also having a regulatory significant and paramount importance, because as I mentioned in my first slide that when you move from toxicological studies first in humans phase 1, phase 2, phase 3, though we are doing the efficacy studies, but at the same time tolerability and safety evaluation, whether it is a uh, adverse reactions called suspected unexpected serious adverse reactions, suspected unexpected serious adverse reactions, adverse reactions, serious adverse events. All these things will be classified as per the guidance given in the pharmacovigilance. You have to make a report like this. The first signal, for example, whether it is a, say for example, it is a fatal, life threatening, you need to report to the agency within 7 days. Okay, non-fatal, you need to report is within 15 days. So there are some a kind of periodic update to the safety update reports, quarterly, half yearly, sometimes 70 days or 60 days. There is a guidance actually in the pharmacovigilance safety in the um, ICH guidance documents, periodic benefit risk evaluation reports, periodic safety update reports, periodic adverse drug experience reports, addendum to them, clinical expert statements, summary bridging reports, PSER, periodic, reports. So all these things will be, one has to capture all the safety data, particularly in the beginning of the phase 1, phase 2, phase 3, even after the NDA approval also. immediately when you submit the new NDA, 4 month safety update. Then onwards, annually. So first of all, this is very important. For example, in the case of in 2020, when the COVID vaccine has come to the market, people that time we don't have any extensive data available to the FDA, so they just release the vaccine to the market because of under the emergency use of authorization. We don't have any much data, right? So now, over a period of time for 4 to 5 or 7 years, we have extensive data how the people are feeling. Maybe it's giving a clotting to the humans or kind of side effects. We have to capture everything in the form of a structured format as per the ICH guidelines called as a pharmacovigilance, periodic safety update. Accordingly, they will modify the patient information leaflet. There's one called, whenever you take any bottle, the bottle, there is a called patient information, physician, sample or whatever patient information leaflet. What is the dose, adverse reactions, and everything will be, they will update the label as per the periodic update of the safety. The development of safety update report during the phase 1, phase 2, phase 3, periodic benefit risk evaluation report after the NDA, that is called post commercial activity life cycle management. Periodically, whenever you gather the data from the market, safety from the clients, from the different agencies, you just try to collect the data, and update to the agency. So all these are whatever is available. I just captured from the available public domain sites. Now, clinical trial operations. How they are going to carry out the clinical trials? First of all, site identification, site study activation, patient recruitment, enrollment, protocol as per the protocol, whether they have meet the criteria of the exclusion criteria or inclusion criteria, investigators brochure, the again investigators CV and everything. So all these things will be, they have to follow as per the clinical CROs. Okay, site management, patient recruitment, clinical supply management, record management. So once the study may be 6 months to 1 year, long-term study or short-term study, maybe particularly in the case of non-clinical toxicology, just only 2 to 3 weeks or 4 weeks only. But in the case of phase 3, is going to become like long-term safety study establishment. That time, that time one has to, once after the completion of all the last visit and last patient, compilation of all the data lock, they call a data lock. Then they do the statistical analytical plan, get it approved by the agency upfront. And as per the statistical analytical plan, they will evaluate all the data, individual clinical study reports, all the subjects, summary of all the subjects, make a compilation of clinical study reports, and then submit to the agency. So in this comes, if you know the medical terminology, medical writing, biostatistics is very, very important. There are so many models actually, so like ANOVA studies, statistical evaluation, how they do evaluation studies, data management, logistics, everything will be comes under clinical operations. So I think with this, I have covered everything. I think I don't want to take much time. I'm just giving one more diagram here. Once again, the same pre-trial, pre-screening, to the subjects, enrollment, informed consent. From the patients, you have to take informed consent. They have to make sure that they are following the same procedure outlined in the protocol. Likewise, in the visit 1, visit 2, visit 5. So whether they follow the same guidelines, these are all, clinical operations actually. So the case study is already completed. So with this, I hope I have covered everything, all the practical aspects of industry. The main point is that you need to know the strategy, you need to know the science, mainly the IND content, that is module 1, 2, 3, 4, impact assessment, change controls. These two are the important features, things one has to follow whoever is going to come to the regulatory, particularly in the clinical NDA. This overall, clinical IND and NDA pathway. Hope I have done everything, considering all the aspects. And if you have any questions, please keep me in the chat box or in the to my email ID, whatever I mentioned in the beginning. In the interest of time, maybe I can take one or two questions. Thank you so much for the opportunity. Thanks to the uh, Sarojini Naidu Vanita Mahavidyalaya Pharmacy Management and Dr. Principal and HOD respected Dr. Mamata for giving the opportunity. Thank you so much.
Thank you, sir. Vinita madam, unmute your mic. Yeah, there are some questions Vinita madam will ask, sir.
Yeah. Good morning, sir. Actually, I am Dr. Vinutha from Department of Pharmaceutical Quality Assurance. Actually, this was a good, very interesting and quite helpful session for us, sir. Thank you for taking your valuable time to give us this session, sir. There are some questions posted by some delegates in the chat box. I'm reading out it for you, sir. So, the first question is by Dr. M. Sweta. In regulatory aspects, how does the OTC pathway differ from the prescription drug pathway?
Yeah, it's a very good question. Actually, OTC also I worked, but OTC means that the label comprehension, self-study, right? Establishment of the safety data and everything will be, established actually. There's a guidelines in the OTC. So by seeing the label, self-study, just by seeing the study, they have to follow complete label comprehension. These two are the main requirement on the counter, study. So before going to the OTC, the mainly it has to be categorized in the FDA site like OTC, like market. There's there are some guidelines. Complete safety is established, right? And understanding by the patient, whoever is going to take the OTC, they have to self-study, self-understanding, self-medication. There's a criteria under the OTC guidelines. So I think that is the main difference. Prescription drugs means that without prescription, mainly it cannot be, the doctor cannot prescribe the as per the, okay, there is a PADUFA guidelines, Prescription Drug User Fees Act. GADUFA means that the Generic Drug User Fee Act. There's a guidance. Likewise, there are OTC guidelines. There's a lot of changes. All these main changes are only once there is establishment of the safety data complete. For example, acetaminophen, Tylenol is no more. But because anybody can go to the market in the, I mean, here in USA, they can get that Tylenol, they can, administer into the, self-study. Because, anybody, the label should be very comprehensive, the dose, warnings, everything should be well defined in the label, self-study, self-comprehension, OTC. There are some specific guidelines in the OTC they have to follow. And one more thing is that how the switch over from the ANDA to OTC, there is one more guidance. The switch over called as a 505B2. There's one more, I think, kind of guidance document is available in the FDA site. Hope I did answer the question.
Yes, sir. There is one more question by Prashanti. Good morning, sir. Thank you for your amazing session. My question is, what are the common challenges faced during NDA submissions?
Yeah, it's a really excellent question, actually. It's a really wonderful question, actually. So the main challenge is that many companies, NDA, right? Particularly, it's a really very good question because I'm thinking how exactly I need to answer. The main challenges are there are two things coming from the phase 1, phase 2, phase 3. Clinical dossier is very important. For example, a company wants to develop and file one drug product, the legacy one. Say, for example, one Pfizer or BMS company, Say, for example, 20 years ago, they developed and they stopped it. Another company, they acquired that, they wanted to further develop and file in the light of new indication, right? For example, okay, Ozempic drug is there. Ozempic is an anti-diabetic drug. For example, if anybody wants to acquire that and wanted to develop for a different indication, because by taking a Ozempic drug, you are going to reduce your body weight, obesity. So it's a two indications. If anybody wants to take that one as this one and go for the submission and filing, FDA may ask you, so do you have enough data to show the long-term efficacy data? That is one of the question. And during NDA, mainly the chemistry manufacturing controls are the main questions actually. Did you submit batch records? Or else did you do any validation correctly? Or the batch suggests whatever you mentioned in the validation, commercial validation? Did you do all the scale up activities? Or why we are not able to see the blend uniformity data? Because NDA first and foremost is that refuse to receive. If you don't file the module 1, 2, 3, 4, if any lacking, they just simply refuse to receive, means that you're going to lose a lot of money, millions of money. People, first of all, they file perfect documents, not missing any documents. For example, in the pre-NDA meeting, right, before going to NDA, first of all, they will see that in the phase 1, phase 2, phase 3, did you ever address all the pre-NDA, pre-IND minutes of meeting points or not? Do you understand what I'm telling? I did submit NDA, right, today. Did I address all the, for the last 4 or 5 years, all the pre-NDA meeting minutes, whatever the FD has asked me, whether I did address all those things, they will first of all see all those things. In my previous company, in the phase 1, they're asking, show the bridging. For example, one drug product or drug substance, which you have evaluated nitrosamine genotoxicity or carcinogenicity in the preclinical pre-IND phase. Did you use the same drug substance lot in the phase 1, phase 2, phase 3 or not? If not, show the comparative impurity profile, show that they are not harmful. You need to make a side-by-side comparison. You need to make a kind of bridging studies. This is very important. You need to carry out the bridging studies or bridging between the non-clinical to the clinical phase 1, phase 2. If you don't show all these things, these are the main important queries receiving from the NDA. And the second thing is that there are very minor called as information requests. For example, some people, they don't want to submit executed batch records. If you don't want to submit executed batch records, hey, we did not find out, we are not able to locate any executed batch records. Can you please send it to me maybe by within 1 week? So as soon as you submit the NDA, information requests are very common. Within the day of filing, maybe 1 week or 10 days, they or 1 month, they keep on asking you a lot of questions, because if they are not able to find out, locate any documents, any simple informations, they're called as information requests. But the major one is called as CRL. They call as a complete response letter. For example, in one company, their content uniformity data is not good. Stratified content uniformity data as per the ASTM guidelines, they are already failed. Okay, everything is not good. They submitted. They're asking, oh, you have a lot of data. I think this is not good. Okay, better to give to CRL. That means you need to resubmit again. Okay, CRL, particularly in the case of like a complete response letter from the agency, means that you need to evaluate, take time, 6 months or 1 year, bargain with the agency. That means your data is not sufficient. Your data is not good enough to substantiate the either CMC, sometimes even clinical also. I think your data is not good. So please, they issue a called CRL. If you get a CRL, oh my God, this is really a big burden on the companies. They need to reevaluate. They have to resubmission called as NDA resubmission. But small things will be information request. I wanted to show some one. I think one. one more clinical. I found out one questions will be, what are the questions will be asking actually? I don't know. Second. Okay. Type meeting review, administrative documents. Are you able to see my screen? Hello?
No, sir.
No, sir.
No, sir.
Yeah, you are not able to see my screen?
No, sir.
One second. I'm sorry. Share, 1 second. Yeah, this is the, yeah. Are you able to see my,
Yes,
right?
sir.
Yeah, this is called PIND, pre-IND, right? Pre-IND meeting, right? Type B meeting. Does the division, see, as I mentioned during the course of the phase 1, phase 2, phase 3, like, type of meetings, type 1, type A, type B, type C, right? So the agency is asking this person, sponsor, does the division agree with the proposed drug substance specification for oxymetazoline hydrochloride, table number 3? FDA response: the proposed drug substance specification appears reasonable. However, final determination in an NDA review issue. Okay. And also FDA also mentioned a kind of statement in the type B meeting in the NDA, like in the phase 1, phase 2, phase 3, in support of your proposal limits on impurities, provide in the NDA information on the impurities as per the current revisions of the ICH guidances Q3A, Q3C, Q3D. Suppose during NDA submission, final NDA submission, if you don't address all the FDA questions and responses, whatever they are given in the phase 1, phase 2, phase 3, they issue CRL. These are the critical things one has to know. I think, do you understand, Adam? That makes sense? Hello? Are you able to
Yes,
understand my question actually? So what I want to say that, all the pre-NDA, pre-IND, phase 1 or end of phase 2, FDA suggestions, responses, we have to address everything in the final NDA submission. Maybe it is connected to CMC, maybe it is connected to clinical. If you don't address, they may ask you in the form of information request or complete response letter, or they may ask you resubmission. There are so many questions you see now. See, does the division agree with the proposed drug product release specification, table number 6, and proposed drug product stability specification? FDA response: the proposed specification provided in the table number 6 and 7 appear to be reasonable. However, for an NDA submission, upon evaluation, the data submitted, additional tests and tightening of the specification may be required. They wanted to tighten the specification. You are giving a blend uniformity 90 to 110%, but they wanted to keep only 95 to 105%. So that you need to address during NDA. Is that make sense, please? Are still any questions?
Yes, sir. Thank you, sir. The question is
yes, no.
answered, sir.
Yeah, because NDA is very critical. Actually, pre-NDA, NDA is during NDA, a lot of questions will be coming because the whole bunch of reports, data, clinical, non-clinical, module 3 and module 1. It's very, very critical because they do multiple times reviews within the regulatory. We have a lot of departments within the regulatory, because I come and maybe I'm overseeing in the global for all the countries, maybe specific country requirement within the regulatory submission management, regulatory clinical, regulatory non-clinical, regulatory operations, regulatory submission management. There are, I think, 7 or 8 departments. They will collect, they will coordinate everything in one meeting. They minimum take 6 months to 7 months to chalk out the strategy, how exactly they need to make a good drafting, to make a layout of all the module 1, 2, 3, 4, 5 to submit to the final NDA submission.
Yeah,
Thank you,
thanks,
sir.
sir. Thank you so much for your informative
I don't know
sessions
how.
here, and mostly useful for us, means for the academics uh, of all specializations of M.Pharm. Actually, it's going to be very much useful. This is so informative and covered all the topics like regulatory authorities of different countries, regulatory pathway means IND to NDA, then CTD modules, clinical trial protocols, means with the help of the formats you have shown, ICH quality guidelines where you focused on Q8, Q9, Q10, and also FDA's meeting management protocol goals. And at last, you covered the pharmacovigilance also, sir. So I think most of the important topics related to this approval process is covered, and we have actually informed quality assurance, regulatory affairs, and pharmaceutics, pharma analysis, pharmacology means all the specializations require these topics, a general topic. And as a faculty means we can understand how much it is there, and we can go through the formats that you have shown. We means we didn't come across these formats, means new formats we can see, which we can utilize giving our lectures also to the students, and what is going on in the market. uh, so in that way, it's very useful, and I request any participants also to speak about this session. Means like it's like a feedback orally, and on the spot you can give.
Yeah, I can, because I love to have more questions, because I wanted to understand where, because I am giving a overall high level and touching the fundamentals. I just to, with a view to disseminate the knowledge and experience to you guys, so I wanted to understand where you are. So please kindly ask questions. Maybe whatever you have it. I mean, I'm allowed to answer. Maybe you wanted to conduct one more session only for the question answer session. I love to have it also. Either way, even now also you can ask questions actually. Maybe one or two questions, then we can wind up maybe typical questions actually, because it's a NDA. Definitely, any NDA will have a lot of questions. There's no doubt about it. But one should not receive the CRL, complete response letter. That is the main important thing. If they, if they accept just to only accept for review, that is a great thing. But during review, generally minor things and a lot of questions, what is the bulk whole time study? Do you have any blend uniformity data, stratified content uniformity data? You submitted the RATS non-clinical data only for 2 weeks study. Okay, for example, in the case of, okay, you did not submit the pediatric study plan called PIP in Europe. In the USA called as a PSP, pediatric study plan. You need to submit even before that, because geriatric and pediatric patients, they need a special kind of the dosage actually dosing requirements. So the toxicology, for example, in the case of one of my in the previous company, they're asking about embryonic rat juvenile animal study. Juvenile animal means that it's only small animals. Fertility study, because mainly nephrology, hepatic impairment, pregnancy, lactation, reproductive, the safety. These are the main things actually for a clinical. For the for the CMC aspects, impurities are very important. Nowadays, elemental impurities, degradants actually, nitrosamine impurities, carcinogenic impurities. These are, one has to address everything will be upfront.
Okay, sir. Any
questions, please,
delegates,
madam?
anyone want to ask questions or speak something? Please. Okay, sir. We'll close the session. No more questions. Thank you very much, sir. And there are around 80 to 90 participants have attended this session, and I think they're going to utilize in their future lectures. You spent
Yeah,
much time for us, sir. It's already, I think, for you, it is the time
yeah.
is
Time is
11 crossed, I think, 11 p.m.
uh,
crossed.
yeah. Then another few hours I have to get up because I have to go to some other meetings early morning. But that's fine.
Okay,
But I'm
okay.
very happy to share all my experience and experience. But I know it's a very high level. But still, if you have any questions, you can send email or chat or whatever in the future. I'll keep in touch also, madam. Thanks for the opportunity. Thanks to Dr. Mamta, madam, for spearheading all the convening a good faculty development program. Opportunity for the international speakers also. This kind of stuff. I
Thank you,
appreciate it.
sir. We are fortunate for that. We have to say thanks.
Yeah, thank you.
Yeah.
Yeah,
Okay, sir.
bye,
We'll
madam.
end the session. Thank you, Mananda.
Yeah, madam. Yeah.
We'll meet again at 11:30. Means at 11:25, we'll join all for one more session. It's by JP Naidu, sir, and the topic is joy of teaching. Okay, see you all at 11:30. Bye.
Okay.